Hello everyone, welcome to session 33. The topic is properties of enzymes. Yeah guys, today we will in this lecture we will try to understand the properties of enzymes. So in the previous lectures we have tried to understand what is an enzyme and what are the components of enzyme, what is the nomenclature and how enzymes are being classified. So followed by today we have to discuss the properties of enzymes. There are some 5 or 6 important properties of enzymes that we are going to discuss in detail. So as we all know that enzyme molecules are very large in size. Their molecular weight ranges from 12,000 to over 1 million. They are therefore very large compared when the substrate or functional group they act upon. It has been observed that molecular weights of enzymes prove to be approximately an n-fold multiples, which is found to be an unit in most proteins. The enzymes possess many outstanding characters. They are, number one, they are colloidal in nature. On account of their large size, the enzyme molecules possess extremely low rates of diffusion. They express very low rate of diffusion and form colloidal systems in water. Being colloidal in nature, the enzymes are non-dialysable, although some contain dialysable or dissociable component in the form of coenzymes. The second important character or property of enzyme is the catalytic nature. Catalytic nature or effectiveness of an enzyme. An universal feature of all enzymes is enzymatic reactions is the virtual absence of any side products. So in an enzyme substrate catalyzed reaction, we expect an intermediate enzyme substrate complex, and this enzyme substrate complex gets converted into enzyme and product. So there are no as such any side products like in chemical reactions. They are recovered as such without undergoing any qualitative or quantitative change. So this is the reason why they are required in very small amounts and are capable of catalyzing the transformation of large quantity of substrates. The catalytic potency of enzymes is extremely or exceedingly great. They have great catalytic potency. The catalytic power of an enzyme is measured by the turnover number or molecular activity. So sometimes it is represented as ETON, enzyme turnover number. The number of substrate molecules converted into product per unit time when the enzyme is fully saturated with the substrate. So this is the definition for enzyme turnover number. The value of turnover number varies. It is going to change with different enzymes and depends on the physiological conditions in which the reaction is taking place. However, for most enzymes, the turnover number falls between 1 to 104 units per second. The third important property of enzymes is specificity of enzyme action. With few exceptions, a majority of the enzymes are specific in their mode of action. Their specificity lies in the fact that they may act on one group, they may act on one group or one specific type of substrate or one group of structurally related compounds on only one of the two optical isomers and only on the two geometrical isomers. So these are the 4 types of specificity which is commonly broadly observed in enzymes. So accordingly, 4 patterns of enzyme specificities have been recognized. So the number 1, it is absolute specificity. So some enzymes are capable of acting on only one substrate. Say for example, urease acts only on urea to produce ammonia, right? So this enzyme is so specific that it is not going to act on any other substrate. It is going to act only on one substrate. So such kind of enzymes we are going to call it as an absolute specificity, right? The second type of specificity we have that we call it, yeah guys, we were discussing about group specificity. So in group specificity, uh, enzymes are capable of catalyzing the reaction of a structurally related group of compounds. Structurally related group of compounds. Say for example, lactic dehydrogenase or LDH. This enzyme can catalyze the interconversion of pyruvic and lactic acid, both pyruvic acid and lactic acid. So this is called as group specificity. Though the third type of specificity is optical specificity. So we all know what do we mean by optical isomers. So we have D and L kind of isomers. The most striking aspect of this kind of specificity of enzymes is that a particular enzyme will react with only one of the two optical isomers, either D or L. Say for example, arginase is an enzyme that acts only on L isomer and does not act on its D isomer. So this is an example for optical specificity. So if we have one enzyme, it is acting on only one type of the isomer, optical isomer of that compound. So we know that we have D isomers and L isomers. So optical specificity is here the enzymes are acting on only one type of isomers of that compound, not on the other type. And then lastly we have geometrical specificity. In geometrical specificity, some enzymes exhibit specificity towards the cis and trans forms of a compound. As an example, fumarase is an enzyme that catalyzes the interconversion of fumaric acid and malic acid. It does not react with malic acid, which is a maleic acid, which is the cis isomer of the fumaric acid, or with D-malic acid. So here in the previous one we have focused on the optical specificity of the enzyme, and in geometrical specificity we are focusing on the cis and trans forms of specificity of the enzymes. The degree of specificity of enzymes for substrate is usually high and sometimes absent. Proteolytic enzymes catalyzes the hydrolysis of a peptide bond. Many proteolytic enzymes like pepsin, trypsin, chymotrypsin, they catalyze different but related reaction, namely the hydrolysis of an ester bond. These enzymes, say for example, I can take chymotrypsin, preferentially splits peptide bonds in which the carboxyl group is from an aromatic amino acids. Yeah guys, now continue with this fourth property of enzymes, that is active site. As the substrate molecules are comparatively much smaller than the enzyme molecules, there should be some specific region or site on the enzyme for binding with the substrate. So actually that site we are going to call it as an say if this is a substrate molecule, and if this is an enzyme molecule, there should be some area where the substrate is going to bind. So that area, this particular cleft or a pocket, we call it as an active site where the substrate is going to bind. So the active site occupies a relatively small portion of the enzyme molecule. The active site is neither a point nor a line or even a plane, but it is a three-dimensional entity. I repeat it, it's a three-dimensional structure or a three-dimensional entity. It is made up of groups that come from the different parts of the linear amino acid sequence. Yeah, guys, we should be aware that so proteins, they can be in a linear chain, they can be in the helices, or they can be sheets, or they can be in the tertiary configuration, or they can be in the quaternary configuration. So the active site is a three-dimensional structure, and the amino acids can orient in different way. Say for example, in lysozyme, it has subsites in the active site. The amino acids that are present in the active site are number 35, number 52, amino acid 59, 62, 63, and 107. Usually the arrangement of atoms in the active site is well defined, resulting in a marked specificity of the enzyme. Uh, the active site binds the substrate molecule by relatively weak forces like the van der Waals forces, or sometimes even hydrogen bonds. The active site in the enzyme molecules are grooves or crevices. The active sites are grooves or crevices where yeah, so they are the grooves or crevices from which water is largely excluded. It contains some amino acids such as aspartic acid, glutamic acid, lysine, serine. These are the most common amino acids that are possibly present in the active site, or in the pocket of the enzyme, that is active site. The side chain groups like COOH, NH2, CH2OH serve as catalytic groups in the active site. Besides, the crevice creates a microenvironment in which certain polar residues acquire special properties which are essential for catalysis of the substrate. Yeah, guys, this is I have taken this image from University of Waterloo. Uh, yeah, this is an example of so this is a large molecule which is an enzyme. So this whole thing is an enzyme, and this area is the active site, or this is the pocket. So this is the active site of the enzyme chymotrypsin. So in the active site, or this pocket, we can see the arrangement of some amino acid chains. Say for example, this is serine 195, it is here, and histidine, which is blue in color, it is here, and aspartic acid, it is 102, it is here. The active site of this it is containing serine, histidine, and aspartic acid residues. So this is how the active site of an enzyme looks like. Now to explain the how the enzymes are going to work, two models were being proposed. Very two popular hypotheses or two theories were being proposed. The one theory is the Fischer's lock and key mechanism and hypothesis, and the other one is induced fit theory proposed by Koshaland. Yeah. So this model is also known as a template model, or it is also called as a lock and key model. It is also called as a lock and key model. So here the enzyme is compared to a large lock, and this is the pocket, or this is the active site of the enzyme, which is a rigid structure here, and the substrate comes and binds into the perfectly. It fits like a how a key is going to fit into a lock exactly. So here the structure of the active site is rigid, so and perfectly the substrate should come and fit into the active site, and it should get interlocked. Hence this theory is also called as a reciprocal fit between the molecular structure of the enzyme and the substrate. The enzyme substrate complex is highly unstable, and almost immediately this complex decomposes to produce the product and the free enzyme. Now let us come to the drawback of this theory. So the enzyme substrate union results in the release of energy. It is this energy which is in fact raises the energy level of the substrate molecule, thus indicating the active site, activated state. In this activated state, certain certain bonds of the substrate molecule become more susceptible to cleavage, and catalysis is going to occur. Now let us look into the this is a other representation of the lock and key theory versus the induced fit. Unfortunate feature of Fischer's model or the lock and key hypothesis is the rigidity of the rigidity of the active site. It is the drawback, or we could not explain. Okay, most of the enzymes are exactly complementary to the substrate. Active site of the enzyme is complementary to the substrate. So this is one drawback of this theory. Now let us look into the Koshaland's induced fit model. So Koshaland presumed that the enzyme molecule is does not retain the original shape and structure, but by the contact of the substrate induces some configurational or geometrical changes. So once the substrate is going to bind to the enzyme, there are some changes in the active site of the enzyme. Consequently, the enzyme molecule is made to fit completely the configuration and active centers of the substrate. At the same time, other amino acid residues may become buried in the interior of the molecule. As to sequence of events during the conformational changes, three possibilities exist. The enzyme may first undergo conformational changes, then bind to the substrate, or an alternative pathway is that substrate may first be bound and then a conformational change may occur. So this is the second possibility. Both the process may occur simultaneously with further isomerization to the final conformation. So these are the three possible sequence of events that may occur when an enzyme comes and meets the substrate, or substrate meets the enzyme. So either the enzyme may undergo conformational changes, or the substrate may undergo some conformational changes, or when they are bound, both may undergo some isomerization or some kind of changes so as to induce fit. There is a possibility, there is a relaxation for fixing. Koshaland models has now gained much experimental support. Conformational changes during substrate binding and catalysis have been demonstrated for various enzymes such as phosphoglucomatase, creatine kinase, carboxypeptidase, etc. So these are my references. Hope you got my point. So in today's class we have discussed about we have discussed about the properties of enzymes. There are 5 important properties of enzymes are enzymes are colloidal in nature. They have specificity. The specificity can be group specificity, absolute specificity, or optical specificity, or the specificity can be geometrical. Then we have discussed about the active site property of an enzyme. The active site is nothing but it is a crevice or a groove, which is a 3D structure made up of some amino acid residues. This is the site where the catalysis is going to occur. So to explain the active site and the mechanism of catalysis, two models or two hypotheses were being proposed. One is called as Fischer's lock and key model. So this is a rigid model, and the second one is a induced fit theory, which was proposed by Koshaland, and this is a flexible model where the active site is not rigid, it is flexible, where there is a room for certain changes which can occur in the active site of the enzyme, so as to induce fit. So thank you one and all. If there are any questions please write in the comment section. I am trying my best to reach out my students during this pandemic. Thank you for watching all. I hope this presentation will be useful to the graduate children. Thank you one and all.